Well, hello and welcome to our Hearing Health Hour webinar. Thank you for joining us for another research presentation from Hearing Health Foundation. Today's topic is a hot one: inner ear hair cell regeneration. Now, these hair cells, in contrast to those on top of your head, are much more important as they make one of our most important senses, hearing, possible. This event has a live captioner. You can enable closed captions by clicking the CC button in the toolbar at the bottom of your screen. If you need any other assistance using Zoom, follow the link to the technical guide shared in the chat. Now, by way of introduction, my name is Dr. Anil Lawani. I'm a professor and vice chair for research in the Department of Otolaryngology, Head and Neck Surgery. You can practice that with me, otolaryngology, head and neck surgery, as well as Associate Dean for Student Research at Columbia University Vagelos College of Physicians and Surgeons in New York. I'm also a board member at Hearing Health Foundation, where I oversee the Emerging Research Grants Program, also affectionately known as ERG. ERG provides critical funds to researchers studying for hearing and balance conditions. And these grants have made it possible for many leaders in our field to become successful scientists, even our own speaker today, Dr. Hartzano. Today, we will hear from two individuals from Hearing Health Foundation's other major research program, the Hearing Restoration Project, or HRP. The HRP is a consortium of amazing scientists working collaboratively to advance cures for hearing loss and tinnitus. Our presenters today are the HRP Consortium Scientific Director, the one who leads it all, Dr. Lisa Goodrich of Harvard Medical School. And I want you to know that I've known Dr. Goodrich since, boy, she was so young, but many years ago, when she came to our lab and we worked together to dissect cochleas. And of course, she's just an amazing scientist, and you're going to hear all about hearing restoration, as well as one of our other consortium members, Dr. Rana Hartzano of the University of Maryland School of Medicine. Dr. Hartzano, as I mentioned earlier, received ERG funding as well from HHF in 2009 and 2010, making me an honest broker of saying that ERG creates amazing scientists. Together, they'll discuss the biology of inner ear hair cell regeneration, the HRP's current work, and the direction of future research. HRP's work is only possible through the generosity of supporters like you. And if you'd like to support our work on hearing loss, tinnitus, and related conditions, you can do so today at hhf.org/donate. Now we'll move to the more important part, the presentation. The present and future of inner ear hair cell regenerations. And please do hold your questions for Drs. Goodrich and Hartzano until the Q&A session. You're welcome to add them to the chat session. Thank you. Dr. Goodrich.
Great, thank you. Let me get here. Okay, are we all good there? So thank you so much, Anil, for first of all, welcoming me into your lab all those years ago when I decided I wanted to learn about the ear, and for all the work you do with the Hearing Health Foundation. It's really much appreciated. And I'm really happy to all of you for being here and listening to what we're trying to do with the Hearing Restoration Project. So just to tell you a little bit about myself, I'm a basic scientist. I've been interested in for a long time in just how do you build a nervous system. It's something really I was fascinated by as a child. And I came into the auditory system through my training. And I have to say, it's just been a wonderful community of researchers to be a part of. And it's because it's interesting problems, and we really want to help people to hear better. And having a combination of those things is really fulfilling. So I'm also happy to be here with my friend and colleague, Dr. Rana Hartzano. And she will provide kind of some useful clinical balance of the things we're doing, basic science, and she of course does wonderful basic science as well. And it kind of speaks to the way this consortium works, where we all work together and learn from each other. So let's start by talking about the sense of hearing. And this is something I think we can all appreciate how much it brings to our lives. So from an animal's point of view, of course, it's absolutely essential as a way of finding your food and avoiding being food. But as human beings, our sense of hearing brings us really so much more. So it helps us to communicate with each other across great distances. Through the phone, we can communicate emotion, we can communicate everything that's happening in our lives when we've been separated so much, for example. And we can also use our hearing just to enjoy the beauty in the world, the natural world, the musical world. And amazingly, we can do all of these things even when there's a lot of sounds in our environment. So we can go to a group and be surrounded by our friends, and hopefully we can do this more as the pandemic winds down. And we can somehow listen to the conversation in front of us, even when there's music playing in the background. And we can extract information from that tells us something about the way the person is standing in front of us. And so how does this work? How are we actually collecting these sounds and turning them into something that has so much meaning for us? So it actually all starts with movement of air. So sound travels as wavelengths, and that's moving the air. Okay, so it's a mechanical thing that has to happen first. And our ear is an amazing machine that collects this mechanical energy and turns it into a signal that the brain can understand. So it starts with the ear, the wavelengths of sound traveling through the ear canal, and then causing vibration of these tiny little bones, shown here next to a dime, these tiny little middle ear bones that are moving because of the movement caused by the sound. So those bones sit here, and then that will cause vibrations that are detected here in the inner ear by the cochlea. So the cells that actually make this happen are the ones that Anil referred to, and those are the hair cells. So why do we call them hair cells? Well, it's because of these little hairs that protrude from their surface. And these are basically movement detectors, okay? When the wavelengths of sound move the bones in the ear, this transmits into vibrations inside the inner ear that activate these hair cells. These hair cells will be activated by mechanical force, but then they actually will turn it into an electrical signal. So here's what they look like inside the ear. And you can see that they can't do this all alone. You can see them here as well, surrounded by all these other cells. These other cells, shown in yellow, are the supporting cells. And supporting cells are really just as important. They shouldn't be relegated to supporting status. They're part of this whole wonderful machine that has to vibrate in response to those sounds and then cooperate to keep the hair cells active and alive and to transmit those signals that will go to auditory nerve fibers and on to the brain. So when the system works, it works beautifully, but there are so many moving parts that, of course, unfortunately, there are many ways that the system can stop working. So there are many causes of deafness, from genetic mutations that might keep the hair cells, for example, from detecting the sound to begin with. There are also mutations that could affect the supporting cells, and there are mutations that can affect many other cells around them. There are also ototoxic drugs that could affect the survival of the hair cells, and many other reasons why the ear might stop working. But one interesting thing is that it very often converges on the hair cells. So many forms of deafness will have a hair cell pathology associated with it. So when we look at a healthy ear, we see all these hair cells. You can see what happens in a damaged ear, either with too much noise, for example, or an ototoxic drug, you actually lose these hair cells. And this ends up being a pathology that's associated with many forms of hearing loss. So right now, how do we deal with this? Well, we need to overcome the loss of the hair cells. And traditionally, we've done this with things like hearing aids, which basically increase the ability of whatever cells that are remaining there to detect the sound. Or we can use cochlear implants, which are a really wonderful way of working around the absence of the hair cells by directly stimulating those auditory nerve fibers. Well, I think all of us right now can appreciate how quickly biotechnology is moving. And in the 21st century, we actually have a lot more options for ways to deal with the loss of hair cells. So For one thing, we have a much better understanding of why the hair cells die to begin with, and we are learning ways to prevent that from happening, and that would be ideal. So, not listening to as loud sounds, for example, but also finding ways to prevent the effects of ototoxic drugs. In addition, we've made great strides in gene therapy and starting to find ways to replace the genes that are mutated in various forms of congenital deafness. What I want to talk to you about today is another approach that we're developing through the Hearing Restoration Project, and that's hair cell regeneration. And that means those hair cells that are missing, let's see what we can do to put them back. So this is not a crazy idea because it happens. So it happens in chickens quite naturally. So this is what a chicken cochlea looks like. Again, they have hair cells, completely analogous to the cells that I showed you before from the mammalian ear. And after noise damage, those hair cells are lost. But remarkably, 10 days later, you'd never even know something happened. It's completely repaired. It's much like if you get a cut on your skin, you've got a scar for a little bit, and then it heals and it goes away. So this can happen. You can rebuild something as complicated as the chicken cochlea. And in fact, the same thing happens in zebrafish. It does not happen in the mature human ear, and it does not happen in the mature mouse ear. However, we actually, through the work we've done, have found circumstances in which you can see signs of regeneration even in the mammalian ear. And that's really encouraging. So while it doesn't happen regularly, there's some low-level spontaneous regeneration that can happen in the vestibular system. In addition, you can also see during the very early stages of life that some of those, an injury can lead to replacement of the lost cells with new hair cells. It happens at a much lower level than what happens in birds, frogs, and fish. And this capacity is mostly lost altogether from the cochlea before the animal can hear. So it might happen in the first week of life in a mouse, for example, and then it's gone. Still, this is really encouraging because it tells us that the cells have the capacity at some point of regenerating. It tells us that all the genes you would need to regenerate are there, and that we can, there's the possibility of putting together something as complicated as the mammalian cochlea again from the parts that are left. So let me tell you a little bit about those parts and what's actually happening, because it informs a lot of what we're trying to do through the Hearing Restoration Project. So this is a diagrammatic view of what's happening kind of inside the cochlea. The hair cells are shown in purple. The supporting cells are shown here in gray. So when there is a damaging stimulus, those hair cells die. They are extruded from the surface, and they can actually send signals in the form of molecules that are detected by the supporting cells that basically signal that something bad has happened. And the supporting cells in a regenerating ear, they know how to deal with this. They hear the alert and they come to the rescue. So what do? Well, they actually will divide, and they divide just once, okay? And they divide, limited amount of time, and they will produce a new hair cell, and then they will replace themselves. And through this, we now can rebuild the cochlea, or the organ of Corti, which is where the hair cells are embedded, exactly the way it was before. You've replaced the hair cell, you replace the supporting cell. In other circumstances, the supporting cell might respond to the signal from the dead hair cell by actually turning into a hair cell itself. And that is another form of useful regeneration. So this is what can happen in the chicken cochlea or in the fish lateral line, but it doesn't happen in mature mammals. So this is something we would really like to understand. What is the difference? Why is it that in some species this happens and not in others? And if we can identify, say, the signals and how the cells respond to the signals and figure out what's different about that, maybe we can come up with ways to overcome this block and basically trick the cells into thinking, for example, that they are back in their youthful days in that first week of life when they were able to do this unprompted. So this is a hard problem. I can summarize it, in a few words, but let's just be honest about what we're asking in terms of what needs to happen to make regeneration happen. Let's consider an analogy of a place like Manhattan. Okay, so Manhattan relies on a very complicated infrastructure. You can think about all the different things that have to be happening to keep the city functioning. You've got the water system, you've got transportation, power, all of it has to work. And now say there's a natural disaster. If a blizzard or a flood came, all of these systems would be impacted in certain ways. And you would need to coordinate across those different groups in order to get the city back to its functional self. Likewise, if we're going to repair the ear, we need to figure out all the different events that happen and coordinate among them. And for this to happen, we actually need to coordinate ourselves. Understanding any one of these events can take a lifetime in one lab. If we really want to figure out a way to regenerate hair cells, we need to do better than that. And we need to work together and coordinate across species, across cell types, across systems, and really push innovation. So that's where the HRP comes in. So the Hearing Restoration Project is a consortium. And this is a group of 13 researchers at universities across the United States and Canada. And the way we differ from other scientific groups is that we are cooperating in a very free way to try to get our science to move forward collectively, where we focus on studies that really leverage the power of collaboration. We prioritize any kind of project that is best done by a group of people versus anything else we can do individually in our labs. We also have a high priority on creating tools and resources that will accelerate research, not just by these 13 labs, but by anybody who has an interest in this. And today, Dr. Herzano will show you one example of a tool that we have supported the development of that I think is a just wonderful example of what we're trying to achieve. We also have a big priority on innovation, where we're willing to just try things that maybe sound, like we're not sure if it will work, but it's worth giving it a try because the payoff is so great. And we have a system that promotes cross-fostering of data and ideas early and often, where we meet very regularly, and we make sure that actually through some of these tools that are created, we can share the data well in advance and not wait around for all these other things to happen before we can actually make use of that data. So these are all the members of the consortium, and it is truly a privilege to get to work with such a wonderful group of people who are all brilliant in the science that they do and thoughtful and generous in the time they spend with each other and with the members of their labs that they are training, who will also go on to do wonderful science. And I think you couldn't ask for a better group of colleagues to try to make something really special happen. So as a group, the HRP has come up with our strategic plan to see how we can collectively advance efforts for hair cell regeneration. And our first goal is to catalog the expression of thousands and thousands of genes across cell types, hair cells, supporting cells, and across species, mouse, chicken, zebrafish, to find those pathways, those sets of molecules that are associated with regeneration or its failure. For example, what are the molecules that make supporting cells in a regenerating species know that they have to divide and produce a hair cell? What are the molecules that ensure that that hair cell can integrate into the remaining epithelium, for example? So this is basically defining the language that we need for regeneration. Then we can use a mouse model system, for example, and figure out what happens when we manipulate these pathways in the mammalian ear, get a sense of how far can we get. And again, this is a matter of testing things and figuring out what goes wrong so we can make it go better. Okay, so this is an iterative process. As we identify the pathways and figure out how they are acting in the mammalian ear, we can use that information to design drugs that have the same effects. So that would be the ultimate goal. So towards this big picture plan, how are we actually doing this? Well, we support a lot of data collection. Consortium members are collecting huge amounts of data from many species, where each lab is using what they know best to generate data that they can share with everybody else in the consortium, so we can compare basically compare pathways across these systems and understand the problem, figure out what's happening, and then use that information to devise a solution. At the same time, we are invested in generating tools and resources that will allow us to extract meaning from all of this data. That includes the gear, which is shown here, and this is what Dr. Hurtado will present. And this is a database where everybody in the consortium, and in fact, anyone who has data to share, can upload it so people can make quick comparisons on their own. We also have invested in a lot of computational analysis of the data to try to go beyond the biology and use computers to pull out information that you can't get just staring at lists of genes. So I will say I like staring at lists of genes, and I have learned something from it on occasion. And then in addition, we are making and testing prototypes where, as I said, we're willing to just try things, see how far we get, use information about how far we get to go back and inform our research again. Another important part of our model is the teamwork. And that's what I started out with by saying that's how the consortium differs. And in fact, everybody in the consortium is part of a working group that is a smaller collection of people who work together to do the experiments, compare notes, and meet on a regular basis so that everybody's kind of on the same page all the time. And then in addition to that, the whole consortium will meet to share data across working groups and to share ideas across those groups. We also have an annual meeting where we all come together and really think about what did we achieve this year? What do we want to do next? Brainstorm new ways to do it. So we're constantly checking in with each other and making sure that we're doing the work in the best way we can. And we're very grateful to get additional input from a wonderful scientific advisory board who help make sure that we've really thought of everything, and that somebody who isn't down in the weeds but can stand back and look at everything has a chance to weigh in and make sure that the work is going in the direction that we want it to go. So those are kind of the overall goals of our group. And what I want to say is that the future really is bright. I think that, it's quite plausible to say that, yes, we can stimulate hair cell regeneration, and this is a reasonable way to go for eventual treatment of hearing imbalance disorders. Why am I optimistic about this? Well, we are learning a lot about these pathways. We've discovered things already, and we're starting to get a sense of what is stopping or starting recovery. As we learn more, there's just opportunity. And I really think it's just a matter of, continuing to do the work that we're doing, and then we will see and discover these therapeutic targets. When will that be? I know that's what everybody would like an answer. The thing with science is what we can do is just keep doing the best work we can and know that we'll recognize the answer. It's very hard to predict. And I would say we have a long way to go, but we have every reason to be optimistic. And I think we could get there as fast as possible, because every day it seems that somebody comes up with a better way to do science, and we are very quick at taking those opportunities and accelerating our own work. In fact, the things we're doing now are using technologies that didn't even exist when the consortium started. So while it's a complicated problem, and I don't think tomorrow we're not going to have the answer, we're doing all the right things to get to the answer, and I'm really optimistic that we will get there soon. So with that, I just want to say thank you very much for your interest in this. And I'm definitely happy to take questions. But first, what I would like to do is hand things over to Dr. Hurtado so she can tell you a bit more about what she's been doing through the HRP to produce a resource that, as I said, is helping not just members of our consortium, but really anybody with an interest in hair cell regeneration. So I'll give the screen to Rona now, if you're ready.
Thank you, Lisa. Let me share screen. Well, it's great to be here this afternoon and with my colleagues and friends, Dr. Goodrich, Dr. Lalwani, and the Hearing Health Foundation, and you are our supporters. I'm a surgeon scientist, and it's not easy to be a surgeon scientist. It's not an easy career path. It's very busy, And it's also very difficult to convince people that you can do it and that you're a worthwhile investment. And in that sense, the Hearing Health Foundation played a critical role in enabling my career as a surgeon scientist. And you can see here that after getting a small resident grant for 2 years, the Hearing Health Foundation actually funded an idea that I had on how to actually be able to analyze gene expression in different cell types of the ear without using complex models. And this really led to additional funding that allowed me to establish everything I do today. And coming back to HRP as a sort of senior member was a delight. And I think one of the greatest things about the HRP, as Dr. Goodrich said, is the fact that we use this combination of expertise, right? So we use something that is called comparative genomics. This is the ability to compare how genes are expressed or regulated across species to reveal the secrets of hair cell regeneration. And you can imagine that a researcher that is an expert in chick biology or in zebrafish biology, they're different researchers. They grew up in different worlds and in different labs. But having their knowledge in silo is not enough. And just to demonstrate to you how far behind the field was and how close we're now, thanks to the consortium, we all know that all of these species have hair cells and supporting cells, and roughly what do their ears look like. But until now, we were never able to say supporting cell A in the chick is the closest neighbor or the equivalent of supporting cell D in human, or supporting cell F in zebrafish. And now, because we know these things, we can start to build the map and build the blueprint that will allow us to come up with ideas of drugs that we can use to induce regeneration in these species that do not regenerate. And we're very close to doing that. And in fact, one of our strategic plan goals for this year is to come up with a paper that will map the hair cells as far as this goes. But how do you get researchers to be able to see these data across all these species? These are very complex data. These are massive data sets. And this is an example here of a data set from Stefan Heller's lab that shows expression of a certain gene in chick. And you can see that it's expressed in these cells that are hair cells, and different types of them. And a data set from Dr. Petrosky's lab showing that the same gene has a little bit of a different pattern because the hair cells are only this group. And if it's colored red, it means it's expressed. So you can see already there are differences. And this is another data set from Dr. Heller's lab. And this is a data set from Dr. Dabdoob's lab, where it's actually more similar to chick. And the idea is you're seeing that the vast amount of data, and we need to be able to see everything in one page. And what the HRP has been doing, it's been supporting an idea that we had and really allowing us to develop it to support the consortium. The consortium served as a focus group, and then really bring this tool as it's developed at real time before publication and make it a main go-to in the field, which is really neat, because when you think about it and research and in almost anything in life, first you develop something, then you publish it, then people can use it. But with the HRP and the very strong knowledge that this is funded by the HHF, there was not a worry of funding. And we were able, as we are developing this amazing resource, to share it at real time with the entire community and advance discovery. So what this tool is it allows you to put in a gene name, and then immediately it comes up with multiple data sets as you choose. And it shows you the expression of that gene in the different data sets. And there are more than 100 curated data sets. There are all from EAR. There are that are public and are organized according to topic. There are more than 800 data sets, just to show you how many people use it, most of which are private. The tool gives you information about the gene you're looking at. It provides additional information if a gene causes hearing loss in mouse or in human. It will light up a button. And it turns out that there are more than 1,000 genes that when we cause a mutation in them, they will cause hearing loss in mouse or in human. And it's impossible to remember all of these 1,000 by heart. So this is very useful. The tool allows us to learn about any data set, expand our knowledge, go to the publication. It allows us to take any data set, for example, a data set that compares expression in hair cells and supporting cells, and turn it into an instant graph and ask to see genes that are high in one population and low in another, or look at all kinds of other research questions that are important in order to develop hypotheses. We have additional options and a new age of omic gene expression. This is called single cell RNA-seq, where we take the tissues and we now, instead of taking the tissue and sequencing what genes are expressed in the tissue, we now take the tissue and sequence every single cell separately. And in fact, the HRP has been a pioneer of that in the ear. And you can imagine how important that is, because the techniques that were used previously, they were like taking a class that has 30 kids and averaging their score after every exam to determine if a specific child did better. And this technique is like getting the score from every single child separately. And we're doing it at the single cell level for all of the organisms, and really being able to identify all of these very small populations to really find what are the cells that are similar, and are we missing cells that are necessary for regeneration in one species? Do we need to create them? Is it that the cells that are the source for regeneration in one system, they're just not responding to a certain signal? And if that's the case, what drug, based on, for example, drug.gov, can we add? And then we have a team, just like Dr. Goodrich said, that will do that. They'll do that with gene editing. They will do that with trying in drug delivery. They will do whatever is needed. And the tool allows gene browsing and so on and so forth. And the neat thing is it is used now as a catalog in the field. So much so that as of today, we have more than 100 data sets in private and public profiles, more than 1,200 registered users in our of the gear, and more than 1,000 monthly entries for research. An important feature of the gear, and what HRP and HHF did, was really encourage teaching. You know, if you make a tool and you don't teach how to use it, it's not useful. So what we've done is, with the support of the HRP and the HHF, we've been going to every single large ear meeting and doing hands-on workshops and teaching individuals for 90 minutes, for shorter sessions, for longer sessions, how to use the tool that we developed. We have Gear Swag that we give people to take home. We offer private sessions to labs. We offer private loading. We have a help desk. We do all of these things because in the HRP, we think that data sharing is important, not just inside the consortium, but that it is important across the board. And in fact, the papers that we generate are based on data that is not just generated by the HRP, but also by people that from the ERG or any researcher anywhere in the world. Finally, I'm really excited to share that the gear actually did get published. This seminar is very timely. It got published in Nature Methods, a high impact journal, just on June 25, a couple of weeks ago. And I will be remiss if I won't thank my team of collaborators, Anoop Mahorkar and Seth Ahmed. Dr. Ahmed is also a member of the HRP, and he's one of our lead analysts. Joshua Orbis is the father of GEAR. He's the lead engineer. Dr. Song supports the help desk, and I can't say enough to thank the HRP, the HHF, and you, because this has really transformed things in the ear field, and it would have never been possible without your support.
Well, Dr. Goodrich and Dr. Zana, that was wonderful. I especially enjoyed your analogy to New York, where I live, and the complexity of the system, and along those lines, given its complexities, if you can have hair cell regeneration, will that lead to hearing, or do the synaptic junctions, auditory nerves, and central auditory processing also needs to be fixed for a human to hear? And that's from Dr. Daniel Fink. It's a really good question. And you're muted, Lisa.
That's a great question. And it's something I think about a lot, because actually what I didn't get to say is that my lab actually mostly works on the auditory nerve and on the spiral ganglion neurons. And so we have thought for a long time about how the hair cells are innervated by the spiral ganglion neurons. And actually, the supporting cells play a really important role in that, not just the hair cells. And so what I can say is that there's a lot of evidence that in the mouse, when you can kind of create hair cells in a place where they're not supposed to be, the spiral ganglion neurons will follow. And they're pretty good at like sorting out and trying to find a target to innervate. And I think that there's a very good chance that if you made a hair cell in the right place, that it could get innervated, and that the neuron is already connected to the central nervous system and will be there and ready to go. So of course, it's going to depend on why the hair cells are lost and what the state of the spiral ganglion neurons is. But I think that in many cases, yes, I think that they could be connected in a functional way.
Rona, did you want to add anything?
I agree 100%. I mean, we see it also in right there, the early phase of the cochlea is very permissive, and which is a little bit different than what happens to the mature cochlea, which is what we deal with age-related hearing loss. And we know that when we regenerate hair cells early on in the permissive phase, we do get hearing.
So, Dr. Goodrich, I think you also made a very important comment. I'm just going to echo again. This project's called Hearing Restoration Project. It isn't called hair cell regeneration. And what you pointed out is that while the hair cells are important, there are lots of other components of the inner ear that are also very important for hearing, and they all have to be, taken care of and stuff. So I think that was an excellent point that I just wanted to echo again. Kim and Richard also asked whether hair cell regeneration restores all frequencies, and will these hair cells restore comprehensible audibility? It's a nice extension of the last question. That is, once you put everything together, will you be able to actually understand words or not?
Yeah. And again, it's going to be step by step, right? And so first we're trying to get hair cells, but Dr. Hurtado referred to the fact that there are different kinds of hair cells, okay? And hair cells are quite different across species, and species hear from different frequencies, right? And so part of what we're trying to do is to understand not just how do you make any hair cell, but how can we start to generate hair cells that are going to be of, the right type, and eventually to be, just the right frequency. I think, and in fact, Dr. Hurtado's lab has done some really important work along this line in terms of determining different kinds of hair cells. So I would say first things first, let's make some hair cells. And that I think we'll learn through that how perfectly tuned they need to be for there to be a measurable effect. I think that that's something we can guess about, but I don't think that we have any way of giving a firm answer right now.
Great. I'm going to paraphrase a little bit our next question asked by several of our attendees, including Lillian and Karen and others, is let's say tomorrow, Lisa and Rona, you identify gene A. That's going to be the gene that we need to restore. what are some of the impediments to us taking the next steps, whether it's clinical trials or can you talk through some of that? And a timeline maybe that, if tomorrow was the day you identified something, what does that look like? What does the intervention timeline look like too?
Well, it depends. When you say if you identify it, it depends on like what identify means, right? So if it's a candidate, that's one thing. If it's something that you've shown has an effect in an animal model, that's a different thing, right? So if it's just a candidate, if somebody does kind of a comparison on year and says, hey, you should really look at this gene, well, we have to do a lot of due diligence in our science, right? We would go back and we would test it in animal models. We would try a bunch of different ways of manipulating the gene. And if we started to get encouraged, we might start to look for in vitro ways to develop drugs that would do the same thing, or we might think about gene therapy depending on what the gene is. But there's a lot of work you'd want to do to make sure that this is working the way you think it is, and that you're not actually in trying to fix the system, introducing an additional problem, right? So it'd be the same in development of any drug, right? Is that you want to first prove that you can solve the problem, then you want to make sure that in solving the problem, you aren't creating new problems. And so that's why clinical trials take a long time, and we need to collect a lot of data and really see what's happening. I think that the other impediment, and I'd like Dr. Fasano to weigh on this as well, is like from a clinical point of view, is delivering these things. And so that is going to depend again on what it is that you're delivering and how often it needs to be delivered. But the ear does have challenges for delivery, and so that would be another kind of level of development that would have to go through. So maybe Dr. Fasano has other things she'd like to add.
This is great. These are great questions, and it's right. It's fun. We're taking such a complex topic, and we're trying to think about it. And the first thing I think when we think about timeline is how do we bin hearing loss, and to realize that hearing loss, right, is not one disease. And so there's the genetic hearing loss, the child that is born deaf, and they're deaf because of mutation in gene A. So hearing restoration for that child will mean returning that missing gene, or which that is really in advanced stages, and there are several companies, even neighbors of Dr. Goodrich, that are up for clinical trials for these genes. Now, the beauty of it is that it's probably going to work, and the limitation is that it's going to help very few people. Because these individuals that have mutations in the specific genes we're looking at right now, and we're not looking at them at HRP, are very rare. They're a very small proportion of the genetic hearing loss. The second group is going to be, let's talk about age-related hearing loss, which is the most common problem. Every other person age 70 should be using hearing aids. And then the question is, what is age-related hearing loss? And is it a monogenic disease? Is it caused by mutation in one gene? In some people it might be. In some people it might be a mixture of genes. And our hope in the consortium and in the field is that, well, if their ears held for 70 years, maybe if we regenerated their hair cells and they got some new hair cells, then they will hold maybe not 70 years, but they will hold, another 10 or 15 years, and that will give them good enough without having to figure out the gene that is missing. And so just as Dr. Goodrich said, it's a multi-step process, and every step opens the door for something else. So at the level of gene delivery, right, we have 3 teams working on gene delivery in HRP, and it very well may be that some of the things they identify will contribute to how some of the clinical trials are going to change and morph and do better. To Dr. Lalwani's question, identify a gene, it's a great question. I mean, there might be a gene that we could add to the cochlea to prevent the aging. and then maybe we don't need regeneration. Maybe we, then maybe the consortium switches its path to maintaining the healthy cochlea people age 40 and above. So it's just a great journey.
So Dr. Zana, that's a great thing. Some of the people had also asked about, is it possible to maintain hair cells and maintain the hearing? So the answer is clearly yes, that some of the things you may discover, in fact, are preventative as opposed to after the fact and trying to restore something that was lost.
Yeah.
Along those lines, several have asked whether hearing loss is the only thing that we can address through this project, or are there things like tinnitus and hyperacusis or Meniere's disease? If you can take that specifically or broadly, it would be great, either one of you.
I'll start just by saying that that the hair cells in the vestibular system are, very important as well, and that maintains your balance. So yes, those would be one obvious target for regenerating those hair cells. Tinnitus, which I actually suffer from, so I really feel for everybody. It drives me crazy, but that often starts with injury to the periphery, but just restoring the hair cells is unlikely to just make the tinnitus go away, because really that's kind of a response of your central nervous system to the damage that happened. And so there's parallel efforts that are tackling the tinnitus. So, there may be situations, depending on why one has tinnitus, where you could fix both things at once, but I think they are biologically different problems. Rona, would you like to add more clinical?
Yeah. So, right, it's you're right on, and really, one thing that's important to know is it's extremely rare to have tinnitus without hearing loss, right? So when we're born, we hear up to 20,000 hertz. And as we age, we lose the high frequencies because the sound comes into the ear from the base of the cochlea, where the higher frequency detectors are. So you can Google "how old are my ears" and find out how old you are. Hearing loss is not something private for a group of people that have some kind of mutations. Hearing loss, you're human by having hearing loss, and as we age and we have more hearing loss, all of us have tinnitus. And if we take anybody and put them in a complete soundproof room, we can elicit tinnitus in almost everybody. If we just keep them in an anechoic chamber long enough. So to answer your question, Dr. Lalwani, if we focus our efforts on preventing hearing loss, and the HHF has now a beautiful campaign to prevent exposure to loud sounds, we will significantly decrease tinnitus. But just as Dr. Goodrich said, once you have the tinnitus, it's not maintained anymore by the cochlea. It's maintained probably by the cerebellum and the brainstem, and that's a different ballpark.
We have an interesting question for both of you, even though I don't think your talk focused on this, but this is a good question I'm sure the audience would be interested in. Nancy and others ask about stem cells and their role in potentially hearing restoration, hairstyle regeneration, or anything. You can take that question broadly and maybe address that.
Do you want to start this time, or do you want me?
Sure, I can, though I think you work more in stem cells than I do. But so stem cells are a great question, right? And when I started hearing about stem cells, right, what I imagined was that we're just going to be dropping stem cells into the ear, maybe not really pipetting them, but maybe injecting them, and all of a sudden everything is going to grow back. And that's clearly not possible. The fluid milieu of the ear is completely toxic to cells that are free floating, and cells need to integrate. So that's not going to be the role of stem cells. We use stem cells to learn on how to direct the tissue that is there. And there is this assumption that is probably true, because it's true across almost all tissues, that there are what we call resident stem cells in the tissue that are sitting there and are waiting for the right signal. And if we can wake those up and make them do the things we want them to do, then we will be able to unlock regeneration. And so the HRP does a fair amount of work with stem cells, specifically the lab of Dr. Idge and Dr. Hiller. And that, I believe that's much of what we're using it for, but I'll pass it on to Lisa.
Yeah, and I agree. And one thing I want to add is that actually the supporting cells, they're kind of stem cells. Okay, so if you look at the morphology of those cells, if you look at the genes they express, they have a lot in common with a population of cells in your brain that actually produce new neurons when you need them. Okay, there is something about those supporting cells that early on in life, they actually they have a lot of potential. And so one of the reasons why I have a lot of optimism for hair cell regeneration is that it's not just that the supporting cells are conveniently located for generating hair cells. This is kind of what they do, okay? Like this is just getting them to, like relive their youth. This is what they started out doing. So a lot of it is about reawakening that. And if you think about, many of you may have heard about the Nobel Prize winning work where Yamanaka discovered a few factors that you could add to a cell to kind of, turn it into a stem cell. And that has been very powerful. It's the same thing. If we can find just a few factors to convince these supporting cells in a mature ear to become stem cell-like again, more stem cell-like, that I think that that's the best opportunity for using stem cells, is to really leverage the natural biology.
This kind of reminds me, about 10, 15 years ago, I went to a seminar on stem cells expecting all sorts of therapeutic talks. And pretty much 90% of it was on development and embryology. And this is what you're talking about, both of you, which is that the stem cells are ultimately telling us what kind of signals are necessary to recapitulate that initial development of hair cells. And that's where they may be handy. This may be for Dr. Hartzano, and I'm going to make this a two-part question, sort of along the same line. One is, if I've had a cochlear implant, can I have gene therapy or hearing restoration? And number two, should I hold off on getting a cochlear implant, waiting for hearing restoration therapies to come on board? I think several of our attendees have asked questions along this line.
Sure. Right. And I just came back from clinic, and people ask that question every day as they ask that question to you. And the answer is that the short and long answer of it is do not wait. And it's not because we don't think that hair cell regeneration is going to happen. It's because hair cell regeneration will happen, hearing restoration will happen. It's going to be a while before it happens, not at a level of a clinical trial for a specific mutation. And even if it's going to take, in an optimistic timeline, 10 years, these 10 years are 10 years that it's important to hear. And cochlear implants, they don't provide natural hearing, but they provide us with the single most important thing that we do as human species. And they provide us with the ability to talk with other people, communicate, and stay part of society. They provide us with the opportunity to stay in the loop. Now, I'm not talking, right, obviously there are deaf societies that communicate with sign language very well, and they're vibrant and successful. But there are a lot of people that are hard of hearing in the hearing world that don't have that type of support. And we can't afford to wait. But in that vein, I'll answer two other things. One is, will you be able to get gene therapy later? Maybe. We don't really know exactly it depends on what happened with the cochlear implant. But I will say that one thing the cochlear implant does do, it maintains our neurons. It stimulates them with electricity. And if we don't have the cochlear implant and we don't have any stimulation, those neurons that the hair cells or the sensory epithelium as it regenerates will need, those neurons will be gone. And then we won't have something to transmit information. And the second thing to remember is, and why I am so optimistic about hair cell regeneration is, let's think about what the cochlear implant does. Realistically, we use around 8 channels that use electricity, current spreads. And despite that, we're able to understand speech amazingly well. The simulation of the implant is nowhere near refined or unique or elegant to what our natural hair cells do, yet the brain does a lot of work for us and allows us to communicate. For this reason, I think that it almost doesn't matter what type of hair cells will regenerate. Maybe we'll regenerate vestibular hair cells in the cochlea, but if they will respond to sound enough, and they'll be sprinkled along the length of the cochlea, then there's a good chance we're going to be able to detect and understand speech.
Dr. Goodrich, Dr. Arzano, that was amazing. It's really amazing what HRP is doing, what you are all doing and contributing to our knowledge. Our audience, thank you so much for attending this very informative presentations by Dr. Goodrich, Dr. Arzano on Ear Hair Cell Regeneration and the Hearing Restoration Project. Hearing Health Foundation is so fortunate to be able to fund this urgent work with your help, you, the listener, you're our attendees today. Remember, you can donate to our efforts to advance better treatments and cures for hearing loss, tinnitus, any disease related to hearing and balance at hhf.org/donate. Well, thank you again, Dr. Goodrich, Dr. Arzano. Thank you, everybody, for coming and joining us. And we look forward to our next meeting together, and have a wonderful evening.